Welcome to genome annotation, a key aspect of genome biology, BMS 23010A. By the end of this class, students will be able to define genome annotation, understand its importance, learn the key steps involved, and explore common bioinformatics tools used in the process. Genome annotation involves extracting structural and functional information from raw DNA data. It's crucial for understanding gene maps and regulatory regions, connecting raw data with biological significance. The genome annotation process includes gene assembly, prediction, annotation, and experimental validation, each step building on the last to ensure accuracy. Genome assembly involves piecing together DNA sequences to form a complete genome, a foundational step in annotation. Reassembling short DNA fragments reconstructs original chromosomes, with completed assemblies stored in databases like ENA and NCBI. De novo assembly constructs genomes without references, using reads, contigs, scaffolds, and chromosomes to identify novel genes and variations. Map-to-reference assembly uses known genomes to guide assembly, identifying SNPs and small variations, crucial for clinical diagnostics. Pairwise sequence alignment compares two sequences to find similarities, using tools like BLAST to identify shared functions or evolutionary links. Multiple sequence alignment compares more than two sequences, using tools like MUSCLE to infer evolutionary events and common ancestors. Gene prediction identifies coding genes and their functions, using algorithms to scan for open reading frames and regulatory elements. Open reading frames, ORFs, are DNA sequences starting with a start codon and ending with a stop codon, crucial for identifying protein-coding genes. Detecting ORFs in eukaryotes is challenging due to introns disrupting exons. Bacterial genes have continuous ORFs, making them easier to analyze. The annotation of the hemoglobin subunit beta gene reveals promoter regions, start codons, and exons, illustrating the complexity of gene sequences. Gene prediction tools like BLAST and GenScan help identify gene features, though they have limitations. Alternative approaches are often needed. BLAST results show alignment of chromosome sequences from rats and mice, highlighting the complexity and precision required in gene prediction. Finding genes involves integrating data sets and tools, recognizing patterns, and applying biological insight, highlighting the complexity of annotation. Non-coding RNAs pose challenges for gene prediction due to lack of ORFs, low conservation, and complex structures, complicating the annotation process. Consider searching for tools that identify open reading frames to enhance your understanding of genome annotation. Function annotation involves comparing predicted gene sequences with databases like GenBank to confirm functions, ensuring accurate genome annotation. Function annotation helps identify homologous genes. Paralogs, which arise from gene duplication within the same species, may develop new functions over time. For example, the human A globin and V globin genes both play roles in hemoglobin formation but have distinct functions. Orthologs are genes in different species that descended from a common ancestral gene. They typically maintain similar functions across species. For instance, the LEP gene in humans and the LEPI gene in mice are orthologs. Leptin, present in both humans and mice, controls appetite and is linked to obesity. Loss of function in mice results in severe obesity. Predicting protein function involves examining polypeptides, motifs, and known domains, which indicate protein function. Different domains have specific functions. Ion channels allow ions to pass through membranes. DNA-binding domains regulate genes. Kinase domains add phosphate groups, and ATP binding domains bind ATP for energy use. Motifs like helix turn helix, zinc finger, and leucine zipper have distinct functions. They are involved in DNA binding, protein binding, and dimerization, respectively. Eukaryotic genome annotation involves 3 primary steps: detecting and masking repetitive DNA sequences, structural annotation, and functional annotation. Prokaryotic genome annotation is simpler than eukaryotic. It involves identifying repeat sequences, structural annotation, and functional annotation, which assigns biological functions to proteins. Sequence homology-based annotation uses tools like BLAST and HMMER for sequence alignment. Orthology detection tools differentiate orthologs from paralogs, while domain and motif analysis provide functional insights. Protein domains are stable structural components with specific functions. The pH domain, for example, is involved in cell signaling pathways by binding specific molecules. Gene families contain multiple shared domains, while gene superfamilies share few domains but have minimal functional overlap. Identical domains in different proteins allow similar responses through distinct pathways. Gene ontology annotation describes molecular functions, biological processes, and cellular components. Enrichment analysis tools like DAVID and Panther provide insights into gene functions and evolutionary relationships. Experimental validation techniques include functional complementation, protein localization verification, targeted functional assays, and transcription factor analysis. These methods confirm gene functions and interactions. Explore functional annotation tools to understand their workings and applications in genome analysis. A study on gene expression profiles of breast cancers in Indian women provides insights into molecular processes and potential therapeutic targets. The DAVID database offers comprehensive tools for functional annotation, helping to understand the biological meaning of large gene lists. The string database allows users to search for proteins by name or identifier, providing insights into protein interactions and functions. For further reading, refer to the listed textbooks and online sources, which provide comprehensive insights into genetics and genome annotation.